you can give us your name and basically what you do, and that's it basically, and then we'll go into Hugh's talk. So Kieran Mullally is my name. I'm a board member of ISPE. My main interest professionally would be commissioning and qualification, and I've been in Singapore for about 5 years now. So that's me. June, is June there? Do you want to give us a few lines about yourself?
Oh, hi. Name is June. I work for No Deviation. I've been primarily in the CQV. Very nice music going on in the background.
Yeah, just so we've got a good few participants here today, so anyone who's not speaking, if you could just mute your mic, that would be fantastic. Lovely. June, continue there.
Yeah, I don't know, is there a standard agenda? Like how do you introduce yourself? Like I'm currently working for No Deviation, like I say, primarily in the CQV field. So I've been working in Singapore and China for a number of years.
Perfect. Janavi?
Hi, Sharon.
Hi, do you want to give us a line or two about yourself?
So, sorry, can you hear me? Sorry.
Yes, I can, yes. Can you give us a line or two about yourself?
Okay, sure. Yeah, I'm working for PM Group as a senior process engineer.
Lovely. Okay, terrific. Zhang Wei Leng.
Hi.
Do you give us a line or two about yourself?
Hello.
Hello.
Hello, can you hear?
I can.
Okay. I'm Wei Ling. I'm from Tesla Therapeutics.
Very good. Very good. Christina, I assume you're Christina from ISPE, is it?
Hi, Kieran. Yes, this is Christina. I'm just listening in.
Very good.
Taking attendance. Go ahead.
Alex C. Alex C., would you like to give us a line or two about yourself? Okay, you don't have your mic connected, fair enough. Moving on, Shan.
Shan, or I'm confused. No.
Sorry, it only says Shan here.
Okay.
Hi, I'm Shanta.
Yeah, Shanta, go ahead.
Look,
sorry.
Anyway, look, I'll tell you what, we're just gonna we're going to abandon the introductions because the participants are now at almost 40. This has been an incredibly successful
Can I say one thing?
Talk.
Can I say one thing?
Oh yes, Simone, of course, say one thing, please.
So I'd like to say hello to everybody. So it's really nice to see you here on Zoom since I moved from Singapore last October. So I've been for 2 years. I'm very happy to see the technical Tuesday keep going. And I see the number of participants is now 37, much more than compared to the time we've been doing face-to-face. So the digital transformation works really well, I guess, because we didn't have 37 people in a room during the technical Tuesday last year, as far as I remember. So I'm happy to see this number and happy that all you are safe. I'm actually in Italy. I'm happy to hear your voice from everyone. Thanks.
Fantastic. And you're right. The our transformation to online has been incredibly successful. We haven't slowed up one bit. So what I'll do is I'll transfer the floor over to Hugh, and we can get started with his talk. Hugh will give us his talk. We'll have a Q&A session at the end if there are any questions. And then at the very end, we might have a few words from two of our upcoming speakers. I'm not sure if Jeff is online today, but I believe if he is, he might give us a few words about his talk that's coming up soon, and Michael Crispin, who's online, who will give us a little talk about a sentence or two about his talk that's coming up next month. So without further ado, Hugh, if you'd like to take over.
Yeah, hello everyone. My name is Hugh McKee. I'm a process engineer based in Singapore. I've been a member of the ISPE for 20 years. My main experience is upstream fermentation, bioreactors, both in vaccine manufacturing, MABs and bacterial. I'm not claiming tonight now to be a protein engineer or virologist or geneticist. I'm just going to give an engineering point of view of some of the points that people may hear about in the cell and gene therapy world. Go through some of the buzzwords people are hearing. I suppose with Simone on the line as well, it'll be a right opportunity to thank Simone and Elad for making the Technical Tuesdays no PowerPoint Tuesdays. So we don't use PowerPoints on Technical Tuesday. So we're going to try to wing this with a whiteboard and just with notes in the background. I hosted 3 Technical Tuesdays last year, one on process safety, one on Pharma 4.0, and one on hygienic equipment design. And during them talks, we discussed that we would try to build up to maybe doing a Technical Tuesday facility design. And this is part of it. So what I'm going to propose tonight is I'll go through some of the background to cell and gene therapy. And then in the next Technical Tuesday that comes available, which I think is in November, we'll try to go through a layout of a viral vector facility. So I just want to kind of go through some of the basics, and I'm going to just annotate some sketches here, and we can start talking about what's behind cell and gene therapy. One of the things I would kind of have laid out is I just go through the principles, maybe protein expression. I want to go through regards sequencing and when things go wrong in sequencing, and then we're just going to go through the immune system. So how the immune system works has a big engineering effect into CAR-T production. I'll touch viral vector, and then one of the areas I want to kind of make it more interactive, like our introductions, is just talk about the ethics behind cell and gene therapy. So look, we all know the DNA sequence, okay? We're all familiar with the double helix. I suppose the timing of what we have here is that, I don't know if people are aware, but I bring up this. The double helix has been around for, as a model for 50, 60 plus years. So I don't know if anyone's aware, but last Sunday, April 25th, which is called DNA Day. So this paper you see in front of us is a paper from Watson and Crick, and they proposed the model of the DNA structure. And that has a huge impact to what we do in cell and gene therapy. And when we look at this paper, and we just kind of pick up, if I can annotate, we just look at the base pairs that are shown here. So these have critical priority over how proteins are expressed, how enzymes are expressed. So these are kind of pairs made up of cysteine, guanine, thymine, and adenine, and how they express themselves is critical. And we'll do a sketch of it now. So yeah, last Sunday was April 25th was DNA Day, and I've just highlighted one thing in here that not many people are aware about. this paper and what's in crick is that there's a highlighted section here which they give credit to someone else for unpublished work. So it's a bit controversial regards who gets the credit to this model, and there's a smoking gun in the middle of it. But that's not what tonight is about. Sorry, I didn't share the document. So this is the document I was on about. So April 25th was DNA Day. This is the first paper published showing the DNA model. So Watson and Crick are attributed to this paper. Buried in the paper, they've acknowledged that they used unpublished data within their paper. That's just a bit of a side to what I want to go through tonight. So we've got a DNA structure. From the DNA structure, through transcription and translation, we get a protein. Okay, so I'm just going to strike a protein here. Very, very simple, okay? So proteins, enzymes, antibodies, all going through the body. And what is driving the formation of that protein is the gene. So the sequence here of your cysteine, all the base pairs on the DNA, they're driving for the replication, translation, the folding of a protein. But for us tonight,
Hugh, just sorry to interrupt you. Just a question: Are we supposed to still be looking at the DNA paper?
Yeah.
Okay, sorry.
No.
Very good. Thank you.
Yeah, let me bring it up again. The next one, she'll share. Thanks for the prompt. So what can you see in the screen now?
I can see the DNA model.
Yeah, perfect.
Arrow coming from it in your pencil.
Okay, yeah. So what I'm just trying to show here is we have the DNA structure, we have a gene, the gene's expressing a protein, but it doesn't always go right. So the sequencing of these bases, and there's thousands of genes in the body, sequencing of them don't always go right. So let's just rub this out and kind of explain where it might go right. So let's try to do text. So DNA sequence, and this should be very simple. It's only 4 letters. Let's draw somewhere else. I'm going to create a protein called Tech Tuesday. I'm going to go tag it there. So let's just say this is a genetic sequence. And there's a few things that can go wrong with this. Let's draw one that I'm just going to type up a few examples. Right, so what we have here, so we have a DNA sequence, and we've got a protein called Technical Tuesday tag. And that's expressing a protein, or it's expressing an enzyme, or it's expressing some sort of biological material. In this sequence, it's been doubled. And then in this sequence, it's missing. So what does that mean? So that means that this expression to the protein may not happen. So we actually might come into trouble that literally the protein is not expressed here, or it's expressing the wrong protein, or it's expressing too much of the protein. What causes the incorrect sequencing? What causes the expression? It could be that it could be an inherited generic or inherited genetic disease. or it could be some sort of form of DNA damage. So let's just draw the two causes maybe. So let's just say we have a genetic disease, or we have some sort of external DNA damage. So we've got these two sequences here, which are expressing, I'm going to say, the wrong protein. And I'm going to talk about later on about, for example, there's a technology called CRISPR gene editing that can actually remove a gene. So literally, scientists can come along and they can cut out a gene. So let's just hypothetically say this gene, this part of the gene, we can take that out, remove it, and then with the genetic modification, the protein can be expressed. What we can do is we could come along with another DNA, and we could literally paste the DNA in. So we've got a genetic disease, or we've got external DNA damage. What I want to talk about is about how we repair it. So how do we come along and repair this damage? And one of the ways we can repair it is tackling what's causing the damage. So external DNA damage, what's the outcome, or what word do we say signs of that is in cancer, in areas where we have some sort of maybe leukemia, and we can modify the body's immune system to rectify this. So I just want to talk shortly now about, say, the immune system, right? So we've got damage being done. The damage has been has impacted the DNA, and we'll just try to fix that. So let's go through it. So I just want to talk now about the immune system, because it leads on to cell and CAR T. So let's draw some cells, T cells. Let's call this, what we call this one. So let's call this killer cell. We've got killer T cells. Got regulator. What's the other one? Helper cells. Sorry, my computer's frozen. No. So when there's an issue with the DNA, a lot of the time the issue is what we see is we could see issue with cancer cells. So let's just see how the immune, or I just want to talk about how cancer cells are handled, treated by the immune system. So we're going to just put in some cancer cells here. So typically on a T cell, there's a receptor. That receptor binds with a an antigen on a cancer cell. They will bind, and then the killer cell will actually bind with the cancer cell, and then it will literally kill the cell. It will make sure that it can't reproduce, it can't replicate in the body. So killer cells have what's called a cytotoxic effect. They literally kill any of the cancer cells that are in the body. I'm going to put text here now. Let's make that C cell. Okay, regulator cells, regulator T cells they regulate the killer cells and the helper cells. So what regulator cells do is they're just keeping the balance on how many cells are in the system. They actually have no impact on or no effect on the cancer cell. But one of the things that does happen with some diseases is the regulator cell can actually hide and mask the cancer. So it can actually bind with it. The cancer can replicate because the regulator has binded with it, and has no killing effect, has no cytotoxic effect. Helper cells. There's two types of helper cells. There's the first one is the helper cell that produces the antibodies when there's the immune system is producing a response to an illness, an infection, be it viral, cancer. So that's the first type of helper cell that will release antibodies into the system, and then antibodies then will bind with any infection, any. The second helper cell is what's used then to as a memory. It's called a memory helper healthy cell, and that will just stay in the system and for future infections, it will remember what antibody is used to kill the infection. And what I'm kind of leading on to is that if we just clear some of this, is that for some cancers, the killer cells that are meant to kill the cancer, sometimes they don't recognize the cancer cell. So for whatever reason, so for whatever reason, the antigen, which is just a protein, is not recognized by the killer cells. So what we'll do is we'll genetically modify the killer cells to have the DNA and the right receptor to bind with the cancer cell. So let's draw a CAR-T cell, and then we get into CAR-T production. So let's draw a normal T cell. You have a normal killer T cell. It has signals in here internally. So this is a killer T cell, and what we're going to do, what happens in cell and gene therapy is these signals are in the lab are taken out and they're modified to bond with an antibody. So the antibody is specific for to bind and recognize with the antigen which might be on the cancer. So what we're left with is two signals, antibody, and then all this DNA is packaged then into a viral vector. So by doing that, what we've done is we've got the signals that the cell needs to trigger the kill of the cancer cell. Got an antibody that will recognize and bind with a cancer antigen. And what we've done is we've packaged the DNA into a viral vector. What a viral vector is it's a transport cart for the DNA. So the DNA that's been modified here is we have to get into other T cells. We have to get into and then into the body. So that the viral vector is used to transport the DNA. So I want to kind of just move on to some buzzwords. So I'm going to draw a patient. Okay. So a patient. And the patient either has some sort of genetic disorder or has some sort of cancer that needs to be treated with a T cell. So there's with cell and gene therapy, there's two ways of delivering the genetic material. You can do it in vivo or ex vivo. So in vivo means getting the viral vector, inserting DNA into the viral vector. So let's say we have a DNA sequence here, and it's going to be in a plasmid. So just say that's in the plasmid. Sorry, no, for that. Swap that out again. There, come on. We then put the plasmid then into the viral vector. The viral vector then is grown. It's grown up once it has the new DNA. It's grown. It's then clarified, filtered, and then it's injected into the patient. So this is called ex vivo, or sorry, in vivo. And I'm going to move on to the next one. So the next one is ex vivo, where we'll take blood from the patient containing the T cells. We'll isolate the T cells, and then we'll use viral vector again to bring the DNA. So we have a viral vector with the DNA, and that DNA is what we saw on the last slide, signals, maybe the antibody. And then I'm just going to dot this here. So these are unmodified T cells in here, and then we're going to bring that viral vector into the T cells, and it's going to carry this DNA. That whole step is called activation and modification. From here, we then put it into a bioreactor. And expand it, expansion. From expansion, then we'll harvest it. And then it goes back into the patient. This whole cycle, ex vivo, is what's called autologous. So what we've done by taking the patient, we've taken the patient's own T cells and modified that patient's own T cells and then expanded it. So it's all related to the patient. So I'm not coming in with another, there are cases where you might take T cells from a donor, but this here is what's called the CAR-T process. And CAR-T process, because of what's happening up here, is the chimeric antigen. So it's antigen receptor. It's DNA that's been, or it's been modified to go into the virus, and then the virus into the T cell. From an engineering point of view, where the difficulties in this whole process, big kind of difficulties are with the capacity of viral vectors, trying to get enough viral vectors. And the second thing is, if you have capacity for this. So when you're building, when you have a facility, most facilities we're familiar with are kind of facilities that people may say in the pharma world are multi-product and multi-platform. But in traditional pharma, we don't run the facilities in multi-product. We usually do a kind of a campaign basis. But in this autologous CAR-T cycle, we could have many patients in the facility. So I just want to kind of go on to the next one. So I'm just kind of going on to how we can demonstrate that the scale up could be difficult. So let's go on to the next one. So what I have here is, I'm going to share it. Can you share? Cross share. So this is something I read at the weekend. Okay, so let's resume share. So this was on LinkedIn at the weekend, where some scientists here in Singapore and in US and Hugh are proposing that they'll use CAR-T cell therapy for treatment for COVID-19. So what I'm speculating is happening here is that they're going to take the patient's T cells. They're going to modify those T cells with an antigen coming from COVID-19. So COVID-19 is a virus, and then on the outer wall of the virus, there's antigens with which are literally just proteins in a special shape, and they're going to put a receptor onto the patient's T cell. And this is a good example of the whole scalability of CAR-T, in that because it's autologous, every single patient is going to have a dedicated donation of the blood. It's going to have its own incubation of its modified T cells, and then it's going to go back back into the patient. So that's just a side I saw at the weekend. So I know I'm kind of going through it fairly quick, but I'm fairly caught for time. I'm going to stop there and pause this and go back to the whiteboard. I'm trying to think what else was on the agenda. So the other thing was, okay, ethics, and this is...
Hugh, have you gone back to the whiteboard?
Yeah,
Okay, yeah,
just gone. Yeah, I've got my tablet here beside me. So I just want to kind of touch then on the ethics of cell and gene therapy. So I don't know if people are aware, but there's two types of cells in the body: somatic and germline. So with these two types of cells, somatic is kind of a cell that isn't passed on from generation to generation. And if you make a genetic change to your somatic cell, it has no impact to future generations. Germline cells are cells like eggs, sperm, where the DNA has been changed in the germline, and it can be passed on to future generations. So in cell and gene therapy, there's a whole ethical issue is whether that is, I wouldn't say allowed, whether it's ethical to kind of to make changes to future generations without their approval, and also what the impact is long term. So there's one famous case in China called I'm going to probably mispronounce it, but the McKee affair, where literally a scientist was able to modify embryos in the lab. The embryos were modified to be HIV resistant, and they were made to be HIV resistant because one of the parents was, I think he believed, had HIV. When he made that change, there was a whole controversy about, is it legal? What are the implications regards future generations? What's the whole issue regards selected editing of the genes? And that's what I have to say on that. The other ethical issue is regards lifestyle diseases, whether if you have someone who maybe has made a lifestyle choice, and because of that has had a disease, be it diabetes or maybe something related from smoke or alcohol abuse. So that's the brief overview on ethics. So that's my overview on cell and gene therapy. I want to next is first of all maybe next time have a layout ready where we can actually look at a facility and go through it line by line, and that's it. That's about it. Look, I know there's 40 odd people on the call that probably have more experience at cell and gene therapy and these kind of viral vector systems than me. So if there's anything anyone wants to chip in or add, yeah, please do.
Thank you for that, Hugh. I suppose we'll maybe have a couple of questions if people have a few questions for you while I'll get the ball rolling. And we'll take a couple of questions then after that. Won't keep it too long. I'm not sure if you would have an answer for this, but just in terms of, I suppose, again, the engineering point of view, the pieces of equipment that are involved. I mean, I mentioned I'm kind of into seeing Hugh a bit, and we'd be very used to dealing with, bioreactors and chrome skids and all that kind of stuff. Can you give us a sense of what kind of pieces of equipment are involved in the cell and gene production in the cell and gene facility? I assume it's a lot of single-use stuff.
Single-use, small scale. So the viral vectors, the bioreactors, will be the same as more or less we see in the small scale in the say mAb production. Downstream, you would have TFF, you generally don't have any UF because the cells are the product themselves. All the equipment are more or less what we see on a small scale in a process development lab. So one of the things about cell and gene therapy, the building blocks for what's going into the facility is more or less there already. So by that I mean maybe say wave bioreactors already there. I know, say, so for some of the scientists in the lab, they have to do, say, buffer exchange. TFF is ready to go. We have TFS from like the ACTA systems, and then all support systems. You know, fridges, freezers, incubators, isolators. They're all the same. A small bit of purification would have chromatography columns. So the building blocks of the plant itself are all the same. It's just that instead of trying to purify a protein, we're trying to purify a virus. So in the MABS facility, we're trying to get rid of the virus. We're always doing virus inactivation. We're always doing virus filtration. So in the MABS facility, or in the cell and gene therapy, we're trying to purify that virus so it can do its work with the cells.
Thank you. Any other questions?
Or any other contributions? So one of the big things with the Tech Tuesday is I meant to be facilitated discussion. You know, I kind of talk there, but we have 40 people online. If anyone wants to share their experience, this is the forum for it.
I think, Michael, did you have a question, Michael?
Yes. First, thank you for your speech. And I'm wondering that you brought the example with the COVID-19, that it would be possible to do some cell therapy on a virus for many people. Because of what I understand was that cell therapy is an individual special therapy for only one human, for that own DNA, for a specific disease.
So
how can that on a virus like COVID-19?
Yeah, so there's so the big discussion in cell and gene therapy is regards to autologous. So for single people, you've got a single process train, and the big engineering challenge as an engineer is how do you scale up for those thousands and thousands of patients. So hypothetically, if these guys, and I hope these guys have some sort of success, they believe it or not, this same team of scientists had success with SARS. They were able to develop a CAR-T cell that could tackle the SARS disease. So what it is exactly that. What you're saying is that this whole scale-up issue in the industry is the big challenge. How can you get scale up from 20 or 30 patients a year to hundreds of patients to, yeah, exactly, potentially COVID, you're talking millions of people. And what type of facility does that look like? What type of layout of facility? Not everyone has the answers at this scale, but certainly regards some of the scales that are coming along, it is. But yeah, you're 100% right. There's an engineering problem there that if these guys find a solution, what happens next regards to facilities is not known. It's a huge scaling up issue.
Hugh, can you hear me?
Yeah, loud and clear.
Okay. Now, also about this, this is very specific per patient, right? Mm-hmm. So how do you do something like this, which is kind of specifically for one patient? So do you have like a set of equipment, or would one have a set of equipment just for one person, or 10 separate ones for 10 people?
So typically, okay. So typically what will happen is the fast part of the process where your materials are coming into a facility, that's a shared room, shared warehouse, shared
Right.
room. When it gets transfected with the new virus, that's going to be a standalone or a shared room. The big challenge is when you have to expand your batch. That is going to have to be several different incubators, and from a GMP point of view, we're always told to make sure there's no cross-contamination. From GMP point of view, there's no way. But the incubators are even changing now, and the rooms that are associated with them incubators. So tying on to what Michael is saying about, what happens when you have hundreds of people having to go process through the facility, the main equipment that changes and the main orientation and layout is your incubators. And that drives the whole the facility design, and then your materials to your incubators, making sure. So do we have specific equipment per patient? You kind of do, and you kind of need to, because your cells are going to react differently to mine, and we can't be cross-contaminating.
Yeah, exactly. And how do you inactivate this virus, the COVID?
So regards what these guys are doing here, the inactivation. Sorry, I'll just share it again. Just bear with me. So what's being proposed here, or what's come out at the weekend is, and you can see the date. So the new modified T cell will have on the cell wall will have an antibody that antibody will recognize the antigen on the COVID-19 cell. So when the new modified cell goes back into the body, what is being proposed here is that the antigens that the COVID-19 is, the T cells will recognize it, bind to it, and then because they'll have the killer functionality of the immune cells, will then destroy the COVID-19 from replicating.
Thanks, Hugh. Thank you.
Any more questions?
So a quick question, Hugh. It's Miriam O'Sullivan here from Amgen. Just in terms of it's a viral vector, so what's the risk if you're doing this on a large scale of viral contamination?
So there's few things to that. So the viruses are selected to be how are you going to say low risk regards how they express themselves. So they've already been genetically modified so that they're not posing any kind of immune or a pathogenic risk to the body. And then they're selected so that they can actually carry the material. So generally, for the cells that are modified for the CAR-T, it's a virus called lentivirus, and that's selected because it can carry. I'm not going to say carry, but it can pack a lot of DNA into its cell and then transcribe that over. Whereas then what you're putting directly into your body is a nanovirus, and that would be a bit more smaller and can attack or kind of transcribe, pass on the DNA to the body cells. Okay.
I have another question. How long does this process take to create the modified virus or the antivirus?
If you anything from the big thing is Michael, is the expression period, and that's when the cells are growing. So after you've done the DNA transcription and the cells are in the incubator, that could be anything from 7 to 14 days. So as a patient, as a patient, when you donate your blood, so you go into a hospital or to a facility near the cell and gene therapy suite, donate your blood. It's not as simple as then going back in 14 days and then being injected with it. One of the things about cell and gene therapy is that a lot of patients have already been through chemotherapy. They've already been through other medical procedures to try to tackle, whether it's, I don't know, leukemia or lymphoma. One of the things that needs to happen to the patient after you've grown the cells, for them cells then, as them cells are being polished and ready to be transfused back to the patient, one of the key things that has to happen to the patient is the patient goes under chemotherapy again. And what that is doing is preparing the immune system for the new CAR-T cell. So as a patient, I don't want my other immune T cells attacking the new CAR-T cells that come into your body, so you prepare the patient for that as well. But from the whole process could be is greater than minimum 14 days. So you can think about it, that facility that you're growing or that you're building, as you're processing batches every day. You've got batches going out and you've got batches in, so you have a huge facility to cater for it.
That means I have to give individual my blood, and then you will create from this whatever vaccination, and that takes 14 days. But I can use only, nobody else.
Yes. Now there's some cases where there's donors, so your T cells may be donated to another patient.
Mm-hmm. Oh, my T cells are really good, I know. But I don't see, especially for this COVID-19, where we have really, let's say, the whole world more or less waiting on vaccination.
Yeah. So what I wanted to show about, what I wanted to show with the COVID-19 is that when you're doing CAR-T and you have a process, there's a huge scalability issue. Now, that one example I showed you is a huge scalability issue that, and in very early days, so the date was the 21st of April. So.
Mm-hmm. Okay, thank you.
One more question.
Go ahead.
So not on me. I was saying to the floor. We'll have one more question.
Only one more question. That's what he's saying.
Yes.
Yeah.
Okay. If there are no more questions, this is, of course, a talk. Thank you very much, Hugh, for the talk today. This is part of an ongoing series of talks that we'll have this year. We're very lucky and grateful that Michael Crispin, who's asked a few questions today, will be doing a talk at the end of next month, at the end of May, on automation. And perhaps, Michael, you'd like to give us maybe a sentence or two or three just to kind of whet the appetites on what your talk would be about.
Yeah. Thank you first for the invitation that I can do this lesson. What I would like to talk is doing project management and what have impact to automation. There are a lot of decisions to make, and doing that, of course, there can be done also a lot of failures because of wrong estimates in the beginning, and they will have a big factor or impact to the cost later for time. And that is something what I would like to talk about from my experience. I mean, I do this automation more than 30 years already. And I think I have some good points regarding to the right system to choose, or I/O decisions, so input-output decisions, and something like this. General project or project flow decisions. So things which I have seen which can go wrong during a project, and maybe should take care for or learn from in other projects in the next time. And that's one of the chapter. Once the depends on the response, I still have some more later on for other chapters as well, but that will be more automation related.
Very good, thank you very much. And we also have another talk, a kind of a slightly softer talk from Jeff Brown, who I don't think is on the call today. I'm not actually sure what his talk is about. I believe it's on the 30th of April. Is it on Microsoft Word, Hugh? Or
I think it's on PowerPoint and Microsoft Word.
PowerPoint?
Yeah,
PowerPoint
so what we're trying to do, or what the ISPE trying to do in Singapore, we're trying to do a work from home series where different people would volunteer and just talk about different packages. So I'm not an expert, but I talked about Zoom last week, and Pierre talked recently about Excel and some other software package. So we're looking for volunteers to help make a contribution, just talk about anything that helps work from home more productive.
Yes, but for the time being, I think Jeff's, in terms of the schedule, Jeff's talk is on Thursday. Is it the 30th of April? This Thursday?
So, yeah.
I think that's it. Okay, so those are the two upcoming talks. We have Jeff on Thursday, and have Michael Crispin on the last Tuesday of the month, which I think is the 31st of May. And that's it for today. Thank you very much, Hugh. Thank you all for attending, of course, and we look forward to seeing you all again soon.
See you. Thank you for facilitating.
Thank you.
Bye.
Thank you very much.
everyone.
Thank you all.
Thank you from Thailand.
Bye.
Thank you.
Thank you.
Bye.